Hey, this is Mr. Mitchell with another science video. Go ahead and write down the questions. Today we're going to talk about genetic disorders and genetic advances. And you can see some of the topics we'll be covering right here on genetic disorders. Some genetic disorders are caused by mutations. Others are caused by changes in structure or the number of chromosomes. Cystic fibrosis is the first one we'll talk about. Cystic fibrosis is where you have a buildup of mucus. You know that you have mucus going down your esophagus, mucus going into your stomach, and cystic fibrosis is a genetic disorder where your body makes a lot of mucus. And instead of having the airway, I guess it's instead of going down into your stomach, it also includes going down into your lungs. Instead of having a clear airway here, you would have lots of lot harder path in which to travel. And the air passages are blocked literally with mucus. And if you have cystic fibrosis, the mucus fills up in your lungs and it makes it hard to breathe. And the cause of it is that 3 bases are not there from the DNA molecule. You know how DNA has all those adenine, thymine, cytosine, guanine bases. 3 of them are not there, and that causes cystic fibrosis. It causes the recessive allele. I think it's probably the more correct term. Another genetic disorder is sickle cell disease. Now you have normal blood cells which look like this. These are red blood cells. But if you have a sickle cell, it kind of has a U-shaped. It looks like an old sickle. Now, you don't hear of sickles very much anymore, but they look like this, and they were used to cut down wheat way back in the old days. But anyway, these sickle cells do not carry as much oxygen as the regular blood cells, and they have more of a tendency to clog things up, as you can see as an example of these sickle cells right here, which are kind of clogging things up. These sickle cells, it's a codominant trait with a normal allele. And a person with 2 sickle cell alleles will have the disease. And I don't know if you can see it very well here, but this is just a regular old Punnett square. This is one of those genes where you can use a Punnett square to figure out if a child, what the percentage chance of a child having the sickle cell disease would be. These 2 people are carriers of the sickle cell, which means they'll have some of these sickle cells, but not a whole lot. But they have that, their offspring have that 25% chance here that they will have the full-blown sickle cell disease, and the 75% chance that it will only be partially having that disease because it is codominant. Oh, looking at my notes to see if there's anything else on that one. There is not. We'll go to hemophilia. Hemophilia is kind of a common genetic disorder. Someone who has hemophilia, well, their blood doesn't clot very easily. So if they get a cut or a scratch, or in this case a nosebleed, it's very hard to get the blood to stop. And anything that's like a bruise is really a bad thing. I mean, it can cause the body just to swell up because the blood just keeps on, it doesn't heal very easily. So someone with hemophilia would really have to watch the things that have to watch just walking around and bumping into things, and accidents can be a bad thing for somebody who has hemophilia. Now, hemophilia is from a recessive allele on the X chromosome. Yes, the X chromosome. So that makes it a sex-linked disorder. The females are XX. They carry this, but it is the males that are far more frequently having hemophilia. Now, we've mentioned before Down syndrome. In Down syndrome, there is a strong degree of mental difficulties and retardation, and heart defects are common in Down syndrome. People with Down syndrome have more of the 21st chromosome, and here's the 21st chromosome down here, more of the 21st chromosome than they're supposed to. Here you see these pairs of chromosomes, 1, 2, 1, 2, and all the way down to this 21st chromosome here. They have 1, 2, 3, so that have that extra chromosome. And the body just has more to process. So somebody with Down syndrome would have so many characteristics of somebody who does not have one. But the body just can't seem to process that in exactly the same way it does for people who have only the two 21st chromosomes. Well, anyway, kind of have a picture of here about a World Down Syndrome Day and so on. Let me talk a little bit about pedigree and karyotype. Pedigree is a chart or family tree, and it helps geneticists trace inherited traits in humans. For example, here we have a father and a mother, and the father is affected with whatever genetic trait. It could have been any of those we said. But they had a child here which was affected with it, a child here that was not, a child here that was, a child here that wasn't, and a child here that was. So you take who they had children with and so on, and you can see how many had that. And that kind of helps geneticists figure out that and Punnett squares, figure out the probability of someone getting that. Now, it used to be that that's all doctors really had, the knowledge of the Punnett square and looking back in the family pedigree, I guess you might say. But today they have things such as karyotypes. A karyotype is a picture of all the chromosomes in a cell arranged in order. You can do that now. And that's exactly what these are down here. We actually had a karyotype in the last slide where you can look at every single one of these chromosomes and kind of get something from it. Now to us, they kind of just like little worm-like figures with little crosses across it. But scientists who are geneticists can do a little bit more with that to know more than we know. But shoot, we know about the 21st chromosome, about how having an extra one would have Down syndrome. We know what having XY would mean here at the end, or XX would mean at the end, and we know what having XY here at the end would mean. This would be male, this would be female. So we know a little bit just from our quick, brief study of it. So in today's medical age, a genetic disorder can have some degree of treatment. If you have some of these things here, I mean, it does not prevent having a happy and productive life, because there are medicine for mucus in the lungs, there are medicine to help manufacture correct blood cells, and there are ways to make people a little bit healthier and happier than they were. Now let's talk about advancements in genetics. Your DNA is different from everyone else's, unless you're an identical twin. But because of advancements in genetics, DNA evidence can show many things. One thing it can show is selective breeding. Farmers have done this for years. Take seeds from the healthiest of plants, only plant them the next spring. Then once fall comes, save the healthiest plants again, only plant them, so on and so on. And it produces the highest of quality crops. And I mean, this mustard seed plant, from the selective breeding from it, you get all kinds of stuff. Broccoli, cauliflower. Now this takes a while, many years, even generations of selective breeding. Because farmers have been doing this for years, and these things have actually come from the selective breeding of this one plant. So a farmer can really get the best of the best if they kind of have a selective breeding plan, you might say. But this can happen also with animals. Inbreeding. We're going to use this example here in just a second, but say you take the two plumpest of turkeys on a farm and put them together, if they're male and female. They will generally produce a plump turkey offspring, or all the offspring that they have would be plump turkeys for the most part. So you have some evidence of genetic disorders, however, whenever you're doing that. But you can do like with fish here. With fish here, say you take all these fish and you just throw away the ones that have like the marks on them and don't have the pretty flesh here, I guess you might say, and breed these, and then any others that have these marks on them, just cull them as well. Kind of get them out of there also. And then eventually you only have the ones that have the white sides to them. Now you could have done the exact opposite. You could have culled this population and bred them to have the dark sides, I guess you might say. So those are examples of inbreeding. Hybridization. Hybridization is kind of the opposite. It's crossing two different animals to get the best traits of both. For example, crossing corn that has many kernels with corn that is resistant to disease could give you a kind of a super kernel of corn. So that's kind of what that turns out to be. Now let's talk about cloning. To make an offspring with the exact same genes is called cloning. That's complex in animals. You actually have to take the nucleus out of an egg cell. You're taking the nucleus out of an egg cell, and you can see here that you've gotten the genetic material out of the original animal, and you're pairing it with this egg cell that does not have a nucleus, and that kind of the blend gives you an animal with the exact same genetic makeup. And it's kind of a touchy subject because you are indeed making a replica of the original animal. Can you imagine if there was a replica of you walking around? Well, anyway, it's been done before. Goats, sheep, pigs, etc. Don't think we've ever had a human clone just yet, even though some groups have claimed that they have. Let's talk about genetic engineering. Genes from one organism are transferred into the DNA of another. Here we have insulin. You can actually inject this little red gene in here of insulin, and it will attach to this circular piece of DNA called a plasmid. And for all of the offspring, this plasmid stays in there. So that's kind of an interesting way to engineer that. Crops have been injected with genetically engineered genes that make them resistant to cold temperatures. Hemophiliacs have been injected with stuff to help keep them from clotting, from clotting the blood. So there's kind of a lot of exciting possibilities with genetic engineering. That's something called gene therapy, and I don't happen to have it in there, but it's in the book. And scientists have a lot of hope in altering other genetic disorders that we previously mentioned by doing genetic engineering and gene therapy. However, is genetic engineering tampering with nature? Well, sure it is. But the question is whether it's right or wrong. Kind of depends on what the intent is of people in genetic engineering. You could have something like something as harmless as improving the corn from bad-looking corn to healthier, good-looking corn. Or you could do kind of what Hitler did. I mean, you may remember from your history books that Hitler kind of was approving of a master race. So he did kind of like the fish here. He took the people that he did not want in his master race and tried to kill them. You may have heard of the concentration camps filled with people that he and his Nazis ended up killing. So Hitler had wanted genetic engineering on a human scale, and of course that's not something that anybody wants. So whether it's right or not is kind of up for you to decide. Hopefully you can complete the questions at least. Hope you learned a little bit about genetic engineering.